All right, so get this. We are going on a deep dive today into a world that's absolutely teeming with life, but you need a microscope to see it.
Oh, I love it when we get into the microbiome.
Yeah, this one is so cool. We're talking about something called the oligomouse microbiota, or OMM12 for short.
Catchy, right?
Well, it's basically this group of 12 bacterial species that are commonly found in the mouse gut. And
Researchers using them as like a model system to try to understand how the gut microbiome works.
exactly. And you send over this fascinating paper from the ISME journal that really maps out all these interactions between these 12 bacteria.
It dives deep into how they coexist, compete, all the drama that unfolds in this tiny ecosystem.
Oh, yeah. Think of it like a microscopic neighborhood, except all the residents are bacteria, and they are all vying for the same limited resources.
Right. It's a constant struggle for survival down there.
So let's meet the neighbors, shall we? We've got 12 players in this microbial drama, and they come from 5 different bacterial families, or phylo, as scientists call them.
We've got firmicutes, bacteroidetes, verrucomicrobia, actinobacteria, and proteobacteria.
Yeah, don't worry if you can't pronounce them all. It's really more about understanding that there's a lot of diversity, even in this small group.
You've got your fast growers, your slow growers, your specialists. It's a real mixed bag.
It's like a bacterial reality show waiting to happen.
Exactly. And to make things even more interesting, the researchers actually used a kind of family tree based on the bacteria's genetic makeup to see who's closely related and who's more distant.
Oh, so like figuring out who's the gossipy aunt and who's the rebellious cousin?
Yeah, and those relationships actually play a big role in how they compete for resources.
Okay, so we've got our cast of characters, but how do scientists actually study these tiny interactions? It's not like they can just interview the bacteria, right?
Well, they've got some pretty clever tricks up their sleeves. One of the techniques they used is called spent media experiments.
Spent media? That sounds kind of gross.
Well, think of it this way. Imagine you have a dinner party, and each bacteria brings a dish to share.
Okay, I'm following so far.
But instead of eating the food directly, the researchers grow each bacteria in a nutrient broth, and then they take that leftover broth, the spent media, and use it to grow other bacteria.
So it's like seeing who enjoys eating whose leftovers.
Precisely. And it turns out that for many of these bacteria, sharing is not caring. They actually hinder each other's growth when they're grown in each other's spent media.
Oh, so it's a cutthroat competition down there.
Very much so. They found that there's a lot of competition for resources, and some bacteria are definitely more aggressive than others.
Okay, so who are the bullies of this microbial neighborhood?
Well, one species, Enterococcus faecalis KB1, stood out as a real troublemaker.
Oh, what did it do?
It actually inhibited the growth of 9 out of the 11 other strains.
Wow, that's like one bacteria throwing shade at almost everyone else at the party.
Exactly. And they think it's because this E. faecalis KB1 produces these things called bacteriocins.
Bacteria what?
Bacteriocins, they're basically like tiny little weapons that bacteria use to fight each other.
Oh, wow, so it's like a microscopic arms race.
And what's even more interesting is that E. faecalis KB1 was only inhibited by its own spent media.
So it's immune to its own weapons.
It seems that way it could even grow happily in the leftover broth of other bacteria.
So it's not just a bully, it can stomach everyone else's cooking too.
Right, it's a very resilient and adaptable species.
Were there any bacteria that E. faecalis KB1 couldn't push around?
Well, there were a few that seemed less affected. For example, Turrisimonas muris YL45 grew well in pretty much everyone's leftover broth.
So it's like the quiet one at the party that just observes everything.
And what's interesting is that its own spent media inhibited the growth of 3 other strains.
Oh, so it might not be a bully, but it's still subtly influencing the environment.
Right, it's like playing the long game.
It's all starting to sound like a soap opera down there with all these alliances and rivalries.
It really is, and we've only just begun to scratch the surface.
So it's not just about competition, right? Didn't they also look at how these bacteria change the environment itself?
Oh yeah. They also looked at how each bacterium affected the pH of its surroundings.
pH, so like how acidic or alkaline it is?
Exactly. Some bacteria make the environment more acidic and others make it more alkaline.
Yeah.
But what they found is that pH alone doesn't fully explain the growth patterns.
So there must be other factors at play.
Right. It suggests that there are more complex interactions happening beyond just, you made my house too acidic.
So it's like a whole web of chemical signals and resource competition going on.
Exactly.
Okay, so we've seen them duke it out in the spent media arena, but how did researchers figure out exactly what each of these bacteria is eating in this nutrient broth?
Well, they use a technique called mass spectrometry.
Okay, and what does that do?
It essentially allows you to analyze the chemical composition of a sample. So in this case, they analyzed the broth before and after each bacteria had its turn.
Oh, so it's like taking a detailed inventory of the pantry to see what each bacteria is consuming.
Exactly. They could see which nutrients were being used up by each species.
That's pretty cool. So did they find any interesting patterns?
They noticed that bacteria who are more closely related on that evolutionary family tree we talked about earlier tended to have more overlap in their diets.
So the closer they are related, the more likely they are to be competing for the same food.
Exactly. It's like siblings fighting over the last slice of pizza.
Makes sense. So they've got all this data on bacterial diets and interactions. Did they just stop there? Or did they try to build a bigger picture?
Oh, they definitely didn't just stop there. They took all this data and tried to build like a virtual gut.
A virtual gut.
Yeah, basically a computer model that simulates all these bacterial interactions they observed.
Oh, wow. So they could test out different scenarios and see how the community might change.
Exactly. It's like a digital petri dish.
That's a great way to put it. And it allowed them to really delve into the metabolic capabilities of each of these bacteria.
Metabolic capability, so like what they're eating and what they're producing.
Exactly. They found that some bacteria, like Flavonofractor plaudi, YL31, were excellent fermenters.
Fermenters. So like they make things like yogurt and sauerkraut. Well, not exactly, but they do produce these beneficial short-chain fatty acids like butyrate.
I've heard that butyrate is really important for gut health.
It is. It plays a role in things like reducing inflammation and providing energy to the cells lining the colon.
Cool. So we've got some good guys in the mix, but I'm guessing not all of them are so beneficial.
You're right. Some of them are more specialized in breaking down specific amino acids.
So it's like a diverse workforce, each bacteria contributing in its own way.
Exactly. And this virtual gut model helped them really see that diversity in action.
Okay, so virtual worlds are one thing, but did they test these interactions in a more realistic setting?
They did. They went from solo acts to pairs in what they call pairwise co-culture experiments.
Pairwise co-culture, so like a bacterial buddy comedy.
Essentially, they grew two species together and watched how they interacted.
Okay, so a little more intimate than the whole neighborhood party.
Right, and unfortunately, most of these relationships were a bit strained.
Oh, no bacterial drama.
Yeah, the majority of the interactions were either -1 bacteria benefiting at the expense of the other, or neutral, where they basically ignored each other.
So no bacterial romances.
Not really. There was one truly predatory interaction, though.
Oh, predator? Who was the victim?
The predator was unsurprisingly E. faecalis KB1, our bacterial bully.
Of course it was. And who did it pick on this time?
Poor Clostridium inoculum, I46. It seems it was producing something that E. faecalis KB1 really wanted.
Not content with just being a bully, now it's a thief, too.
Well, bacteria are all about survival, so they'll take advantage of any opportunity.
So how did they figure out what E. faecalis KB1 was stealing?
They used a technique called metabolomics, which is like analyzing the chemical soup of the co-culture.
So like seeing what's being produced and what's being consumed.
Exactly. And they found that C. inoculum I46 was making a substance called malate.
Malate, does that ring a bell?
Well, it's a key intermediate in several metabolic pathways, and it turns out E. faecalis KB1 was gobbling it up.
So one bacterium's trash is another one's treasure.
Exactly. This cross-feeding is actually a pretty common phenomenon in microbial communities.
So it's not all cutthroat competition. There's some cooperation going on, too.
Right. It's a delicate balance. And to really confirm this cross-feeding, they use special labeled versions of malate and another potential food source, methionine.
Labeled. So like they put little name tags on them?
Not exactly,
Nice.
but they used isotopes, which are like slightly different versions of the same element that can be tracked.
Okay. So they could see where these substances were going.
Exactly. And they found that E. faecalis KB1 was indeed taking up the malate and methionine produced by C. inoculum I46.
So it was like a little metabolic heist.
You could say that.
Okay, so we've got competition, we've got toxins, we've got some frenemies, maybe even a bit of a black market going on with this cross-feeding. What's next?
Well, eventually had to see what would happen when the whole gang got together.
Oh, the ultimate bacterial house party.
They put all 12 bacteria together in what's called a batch culture.
Did it turn into total chaos?
Surprisingly, no 10 out of the 12 bacteria actually managed to coexist.
Wow, really? I thought for sure it would be a free-for-all.
You'd think so with all the rivalry they'd observed, but somehow they found a way to share the space and resources.
So who are the winners in this bacterial house party?
3 species were particularly dominant. Flavonofractor plaudi, YL31, our master fermenter Blaudia cocoides, YL58, and surprisingly E. faecalis KB1.
Wait, E. faecalis, KB1. Our bully made it to the top.
It seems so. It's a very successful species, even if it is a bit aggressive.
I bet the researchers couldn't resist seeing what would happen if they removed E. faecalis KB1 from the mix.
They created a community without E. faecalis KB1 just to see how things would shake out.
So did removing the bully bring peace to the neighborhood?
Not exactly. It was more complicated than that. Some species like C. inocumi 46, our mallet producer, actually flourished without E. faecalis KB1 around.
Oh, so it was actually benefiting from being bullied.
In a way, yes. And another species, Bifidobacterium animalis wild 2, also increased in abundance.
Interesting. So E. faecalis KB1 was kind of keeping them in check.
It seems, so it's like a delicate ecosystem where even the bullies play a role.
It's amazing how complex these interactions are, and this is just with 12 species. Imagine what's happening in the human gut with hundreds of species.
Exactly. It's mind-boggling, but that's what makes it so fascinating to study.
Okay, so they've got this community of bacteria growing in the lab, but how realistic is that compared to the actual gut environment?
That's a great question, and the researchers wanted to make their setup more gut-like.
Okay, so did they create like a tiny furry test tube or something?
Not quite, but they did start by tweaking the nutrient broth.
Tweaking it how?
Well, they lowered the glucose levels to better mimic the conditions in the gut, and then they added different supplements.
Supplements like vitamins and minerals?
Not exactly things like mucin, which is the main component of mucus.
Mucus? Ew.
Well, it's an important part of the gut environment. It helps protect the lining and provides a habitat for certain bacteria.
Okay, I guess I can see that. So they added mucus. What else?
They also added specific sugars and even starch.
So they were like catering to the specific tastes of these bacteria.
Exactly, and it turns out that certain bacteria really thrived with these new menu options.
Like who?
Akkermansia mucinifila, YL44, loved the mucin, which makes sense given its name.
Yeah, it's like the mucin connoisseur.
And Bacteroides caesumeris I48 was a big fan of the extra sugars, while F. plaudi IL31, our fermentation champion, went wild for the starch.
So it's like they were creating a custom buffet for these bacteria.
Exactly, and by doing so, they could actually manipulate the growth of different species.
So they were like microbial chefs crafting the perfect recipe for a thriving gut microbiome.
That's a great analogy.
But the ultimate test was to compare this lab-grown community to what's actually found in the mouse gut, right?
Absolutely, they wanted to see how well their model reflected reality.
Okay, so did they dissect some mice and take a look?
They did. They looked at the bacterial composition in different parts of the mouse gut, the ileum, cecum, colon, and feces.
Wow, so like a full gut tour.
And they compared that to the communities they had grown in the lab.
They match.
They found some interesting correlations. For example, in the cecum, colon, and feces of the mice, the dominant species were B. caesumeris I48, B. cocades, YL58, and A. mucinifila YL44.
What, those were the ones that thrived with the added supplements in the lab, right?
Exactly. So their recipe was actually working. They were able to nudge the community in a certain direction by changing the environment.
That's incredible. So it really does matter what we feed our gut bacteria.
Absolutely. It has a huge impact on the composition and function of the microbiome.
What about the other parts of the gut?
When the ileum, a different section of the gut B, cocoids YL58 and Lactobacillus reuteri I49 were the dominant players.
And were they able to recreate that in the lab?
They were. By changing the supplements in their cultures, they were able to shift the composition of the bacterial community to better reflect these different gut environments.
Wow, so it's not just a random assortment of bacteria. The environment really does play a role.
It's a dynamic ecosystem, and we're just beginning to understand how it all works.
It's like we've zoomed in on this tiny world and discovered a whole new level of complexity.
And interdependence.
It really makes you think about how much we still have to learn about the gut microbiome, and this is just a simplified model with 12 species.
Imagine the complexity in the human gut with hundreds of species interacting.
It's mind-blowing, but also really exciting because the more we learn, the better equipped we'll be to take care of our gut health.
Exactly. This study just gives us a glimpse into this amazing world within us.
And speaking of glimpses, this study was done in a controlled lab setting, but the real gut is a much more dynamic environment.
Oh, absolutely. There are so many other factors that come into play, like the host's immune system, diet, lifestyle. All these things are constantly changing.
Right, so how do those things affect this bacterial community?
Yeah, it really is like trying to solve a giant puzzle, except the pieces are constantly moving and changing. But that's what makes it so fascinating, right? This whole field of microbiome research is still so new. We're just scratching the surface.
And the study, despite all its limitations, it gives us a framework for thinking about these interactions a starting point.
Exactly. And one of the big takeaways is that we can't just think about good bacteria and bad bacteria in isolation.
Because they all interact with each other.
Right. It's all about understanding the balance, the interactions, how the environment shapes those dynamics.
So it's like an ecosystem. It's not just about finding a magic probiotic.
That's a great way to put it. It's about understanding the ecosystem as a whole.
Okay, so how do we do that? How do we become better caretakers of this inner world?
Well, one of the most important things is to nourish those beneficial bacteria with the food they love.
And what do they love?
Fiber. A diverse diet, rich in fruits, vegetables, whole grains, is key.
So it's like creating a diverse buffet for our microbial friends.
Exactly. The more variety, the better. And I've heard that fermented foods are also good for the gut.
Oh yeah, like yogurt, kefir, sauerkraut, all those tangy things.
They contain live bacteria that can contribute to the diversity of the gut microbiome.
So it's like adding reinforcements to the good guys.
Exactly. And then there are prebiotics.
Prebiotics, those are different from probiotics, right?
Yes, prebiotics are basically food for the probiotics. They help them grow and flourish.
Oh, so they're like the cheerleaders for the good bacteria.
You could say that they're found in foods like onions, garlic, bananas, asparagus.
Interesting. So it's a combination of adding good bacteria through things like fermented foods and then feeding the ones that are already there with prebiotics.
Exactly. It's a two-pronged approach.
But it's not just about what we eat, right?
No, absolutely not. Our lifestyle choices can also have a big impact on the gut microbiome.
Like what kind of choices?
Things like managing stress, getting enough sleep, and being mindful of antibiotic use.
Oh yeah, antibiotics, they can wipe out both the good and bad bacteria.
Exactly. So it's important to use them only when necessary.
So it's really about approaching gut health from a holistic perspective.
Absolutely. Taking care of our overall health is essential for creating a thriving environment for those microbes.
It's like we're not just individuals, we're entire ecosystems.
That's a beautiful way to put it.
Well, this has been an incredible journey into the world of the OMM12.
It really has.
We've learned about bacterial bullies and frenemies.
Cross-feeding and virtual guts.
And the delicate balance of this microscopic world.
It's amazing to think that all of this is happening inside us right now.
I know, it really makes you appreciate the complexity of life.
And the power of these tiny organisms.
And so to our listeners out there, remember, you are not just you.
You are a whole universe of bacteria.
And by understanding and nurturing this inner world, you can unlock incredible potential for health and well-being.
Thanks for joining us on this deep dive.
Until next time, stay curious.